So we're picking up right where we left off, where we're talking about how to make certain amino acids. And actually, we've already gone over alanine and aspartate in particular, and you should know what enzymes are involved and where they come from. So for example, with alanine, what is the precursor for alanine? What other compound is used to make alanine directly? Pyruvate, and the amino acid is, the enzyme is alanine aminotransferase, and aspartate comes directly from what alpha-keto acid? Oxaloacetate, and it's also then aspartate aminotransferase is the enzyme that's used to make it. And so that's all the review for that part that we'll go over. Now we're going to talk about the synthesis of asparagine. Okay, so asparagine, let me get my notes. Given what you know, what amino acid is the originator of asparagine? Does anyone know? What does it look an awful lot like that you think that it'd be easy to make it from? Lysine or glycine? Lysine? Possibly. What else? Is there one that's even more similar to? Aspartate. That would make the most sense, right? From aspartic acid, you're just going to transform it from a carboxylic acid to a what? What's the functional group? The difference between aspartate and asparagine? An amide. Okay? And so then you need to know that what other players, so to speak, are involved. So we're going to start off with oxaloacetate if we're going to work our way in the beginning. So we have oxaloacetate. What compound is compound number 1? No. That's glutamate. And number 2, so glutamate becomes alpha-ketoglutarate. And the enzyme involved in that one is
aspartate aminotransferase.
correct. And so there we go. Oh, whoops. There we go. So then what happens here is aspartate is the prerequisite or precursor for asparagine, and it's glutamine that's utilized. Okay, so glutamine is the other, this is also a ping pong type of reaction. So glutamine becomes what? What's number 5? This is glutamate. Glutamine donates its amine group to become glutamate. Okay, and so what enzyme do you suppose that this is going to be? It utilizes an ATP because it's not it's a ping pong type reaction. So the ATP is used to energy to facilitate this, to make it a good leaving group. What? Say it with some.
Asparagine synthetase.
That is correct. So see, asparagine synthetase. And the reason why it's a synthetase was because it required ATP. It required the energy from the output. So that's why, if you know, as long as you know the classes of enzymes, 99% of the time, well, maybe not quite that high, because there are some of those odd ones like glutaminase and enolase, but more than 90% of the time you'd be able to get it correctly. Okay, so now asparagine can be degraded also to form aspartate. So given what you know about glutamine becoming glutamate, because we actually already did that in a previous chapter, because it had to do with getting rid of nitrogens. What enzyme do you suppose can do this? What was the enzyme that did the glutamine to glutamate? Because it wasn't just normal aminotransferase, since that's not the nitrogen that's being given. Glutaminase. And so knowing what you know now, what enzyme do you suppose is going to be the enzyme that takes asparagine and degrades it to form aspartate? If it was glutaminase, then it would become asparaginase. And this actually has a very unique catalytic triad. Okay, so I want you to know this mechanism. It does go through the same type of idea of a covalent tetrahedral intermediate, but this one actually has multiple triads. So before, when we talked about the serine proteases. Remember, it was like the asp, his, serine for the serine proteases, and it had a cysteine if it was a cysteine protease. But here, we have different amino acids involved. Okay, so this, I don't know why it makes it so small. Here is the asparagine that's going to become aspartic acid, or aspartate. So the first catalytic triad forms the tetrahedral intermediate. And it's going to get rid of, with the nitrogen is going to be released. Similar to what we see with glutaminase. Okay, so the role of the glutamate is really just to take away the hydrogen from the tyrosine. So the tyrosine can take the hydrogen from the catalytic threonine. It's a threonine alcohol that does the nucleophilic attack, similar to the way that the serine did in the serine proteases that we saw last semester. So we have this threonine, which becomes more catalytically active because its hydrogen is being removed by that tyrosine that's close to it in three-dimensional structure. Okay. The other threonine on that other side is just holding the asparagine in the correct orientation. It just holds it in place. Because otherwise, you remember, there's going to be some type of there could be free rotation around that, and so therefore, you wouldn't be able to smack the correct side necessarily. It wouldn't be the right orientation for the catalysis to occur. So that's the second step, is you're going to get collapse. So we see that, just like what we do, whoops, there we go, where we get collapse of the tetrahedral intermediate, and the ammonia, or ammonium, would be released, depending upon the pH. So that's a decent leaving group. But it's still stuck, covalently attached to the original nucleophile, that threonine. We've got to be able to get our enzyme back. That's when the second catalytic triad comes in and it activates a water molecule. So, we can see here, there's the threonine that was the original nucleophile that is still stuck to what will become an aspartate. But it's trigonoplanar. It's not a tetrahedral intermediate. It's an activated water molecule. Let me just change colors. It's an activated water molecule here that's going to go and smack that carbon in order to make this tetrahedral intermediate. And the reason why that water molecule can act as a nucleophile is because of the fact of the other triad. It's not the threonine of the triad that actually does the nucleophilic attack. It's a water molecule. That's what that means. And the reason why is because we have this lysine. The lysine takes away, the hydrogen from the aspartate, holds it in place. The aspartate then is working with the threonine. The threonine is then working with the water in order to make the water more of a nucleophile. And so then, once the water attacks it, we now have another tetrahedral intermediate. We get collapse of that tetrahedral intermediate, and the leaving group then would be the original enzyme. That's what this is showing down here. So we get our threonine back, which is the free enzyme, and we also get aspartate, or aspartic acid, depending on the pH. So that's how we transformed asparagine into aspartate. Even though it's obviously more complicated than the oxaloacetate, just becoming aspartate. Okay. All right, and so this was the final scenario. I had just blown it up. All right, so asparagine metabolism actually has biomedical implications. Just like with many of these things, we've talked in the past about like phenylalanine. We'll talk some about tyrosine, and some of our other classes we've already talked about tyrosine and tryptophan. But asparagine metabolism also has ramifications. For unknown reasons, like childhood acute lymphoblastic leukemia, ALL, it requires the serum asparagine for growth. Okay, it requires extra asparagine. They don't know the reasons why. So if it requires asparagine, given what you know so far, what do you think, what's the type of treatment that you could use for ALL? Where could you try to find a potential drug? What enzyme would you want to possibly target? Asparagine synthetase? Okay. What else could you do? You could target that one. What about, what enzyme could you also give as a treatment? Asparaginase. And actually, they've done asparaginase along with the chemotherapy, and it has more than a 95% remission rate whenever this is done in conjunction. Okay? And only about 10% actually suffer drug resistance. And they are looking at asparagine synthetase inhibitors for possible treatment as well. And so that way you can try to combat it on all sides, hopefully. Chemotherapy to limit the growth of the tumor via, some type of transcriptional control. Asparaginase to degrade asparagine more quickly, and then the asparagine synthetase inhibitors to decrease the amount of asparagine that's present for the cell. The cancer cell, I should say.
Does this only work in children because of the...
I don't know. I have no idea. And I don't know if it's only that this form of leukemia has a high requirement for asparagine, or the reasons why. Why does it need so much asparagine? I don't know. So that's the end of part 2.
I have a question about the mechanism.
So now we're going to start with the third part of this chapter, which is some of the molecules and biomolecules that we have talked about in the past, and some new ones even, that come directly from, or that are derived from amino acids. And I've just picked a few of them, okay? And I doubt that we get all the way through this today. That's not the goal, but we're going to get through part of it today. So, amino acids themselves are often used as building blocks for other things, including hormones, which actually we'll cover towards the very end, coenzymes, nucleotides, porphyrins, which, what is porphyrin? Where have we seen that before? It's in what? Oh, no, that's a porin. What's porphyrin? Mm-hmm. It's hemoglobin. Yeah, that's what makes up hemes. Okay. All right, and antibiotics and neurotransmitters. So let me make sure what order. We're actually going to start with the porphyrin. What do you suppose, which amino acid, if you think about the heme, how big the heme is, what amino acid in animals do you suppose is the precursor for it? Like if you were going to try to design, remember, there are some nitrogens, but it's really carbon heavy. You would think it'd be something that has a lot of carbons in it. It's actually glycine. So glycine just kind of blows your mind. The fact that the tiniest and the most simple is used to make something that's one of the most complex things. Like organically, if you try to do this organic synthesis, it would take a miracle in order for this to occur. Okay? And actually, we already answered that. The use of the porphyrin, you think about the porphyrin ring, that's a cofactor, and that is because it's in hemes. So the hemes and cytochromes, things like that, that will have their ramifications. Glycine actually reacts with succinate. Okay? And so where have we seen succinate? From where can we get succinate? Citric acid cycle. Now, in plants and most bacteria, the intermediate that's formed from the synthesis of the glycine with the succinate is called delta aminolevulinate, and that's formed from glutamate instead of glycine.
It's not intermediate.
What? This delta aminolevulinate is an intermediate. And the iron isn't inserted until the very, very end. Okay. You have to make the protoporphyrin group, which proto is like prototype, porphyrin group before you can actually put the iron in. Okay, so I wanted to show This is the beginning versus animals versus plants and bacteria. And we have an intermediate, but it's directly done with the synthase. I could have asked you what enzyme that was, but I didn't hide it. But hopefully it makes sense to you, the fact that this is delta amino levulinate synthase, because it doesn't require ATP, and it just decarboxylates and coenzyme 8. It removes the coenzyme A from it in order to form the amino levulinate. Whereas it's much more complicated in plants and bacteria. In the event that you have a synthetase, and they actually utilize something that's unique as the way to activate it. Normally we've been using things like coenzyme A or acetyl groups, or many times phosphate from ATP, or even the AMP part, because those are great leaving groups. What plants and bacteria utilize is actually a tRNA. So tRNA gets added to this really tiny glutamate, and then ATP comes in, activates it, and then what happens is you get the glutamyl tRNA reductase at the expense of an NADPH to make the semialdehyde, and then an amino mutase comes in to make delta amino levulinate. So it's much more complicated in bacteria and plants than it is for us. But I think it's really cool that tRNA here is actually used as a leaving group, way to activate the intermediate. Then there are lots and lots of steps. You do not need to memorize these. Okay? I just wanted to show that it's complicated. For us, I would expect you to know the delta amino levulinate synthase. But that's pretty much it, other than the fact that you should know that it's a very, very complicated process. You go through all these pre and pro and proto molecules of the heme before you actually make the heme. And notice the very last step, I do expect you to know the last step is where the iron comes in. So you don't have to worry about iron and the oxidation state of iron messing things up earlier on. For those who have taken neurochemistry, and I think probably chemistry cancer, we've discussed in the past how iron can be kind of finicky based upon its oxidation state. And so therefore you don't have to worry about anything weird happening because of the iron until the very end. For the what?
The enzymes are so bad.
Well, the enzyme names, but if you look at it, it makes sense because they're named after, like, for example, number 5 is coproporphyrinogen oxidase. And that's because it oxidizes coproporphyrinogen 3. And so,
People in this field, that's not.
well, a lot of times they give them, as you know from some of the papers that we read in neurochemistry, they just give them little abbreviations and acronyms, as they call them, instead of saying all of that.
It's so confusing. Remember which.
Right. Remember what's what. Okay. So once again, iron's not incorporated until the very last step. The porphyrin biosynthesis is regulated by the actual concentration of heme, which should make sense. And what happens is it's feedback inhibition. You have a lot of heme around. It turns off some of the early steps. So that way you're not wasting your glycine or your succinate, which makes sense. You don't want to waste away your succinate, since that subtracts from the citric acid cycle, and glycine for that matter, because glycine is utilized for other things as well, not just protein synthesis.
And you said we had to know the slide before this?
Right. Oh, just for humans. I mean, for animals. Yeah. And so, which is just one step. And the fact that the bacteria and plants do it differently, but I'm not going to ask you to memorize all the steps for plants and bacteria, since very few of you are wanting to go into botany or...
Very few.
Well, I was trying to be nice. In this class, none. I have had some people take biochemistry before that were wanting to go not in the biomedical field. All right. There are many, many genetic defects along this way, okay? And there's lots of different diseases. They're called porphyria diseases. You may have heard of some of these. I mean, anytime that you have something so complicated like this, you've got a lot of opportunities for things to go wrong, Okay.
6 anemia.
Yes. And so this is history integration. So King George III, who was the king of Great Britain during the American Revolution, he suffered from porphyria disease, and it causes abdominal pain and neurological disorders, and so that's one thing. He was supposed to be a mad king. Of course, why did a lot of it's not just the British monarchy, but just the European monarchy had a lot of porphyrian diseases.
Because of inbreeding.
Inbreeding, exactly. And so they accumulate these diseases. So you have things like hemophilia. You have different types of anemias, things like that runs in the bloodline there. Okay. There's another rare form of porphyria that results in the accumulation of the uroporphyrinogen 1. So you have too much of one of the precursors, and what it does is it causes your urine to turn red. It also causes your teeth to fluoresce in UV light. Now, I don't know. So they'd be great at a rave. So they are sensitive to sunlight, so they can't be out in the sun for very long. And they have severe anemia. So what would happen if you have severe anemia? What would happen to your skin? What kind of look would you look like? You could kind of almost look blue, or you can look very, very white. And so the ultimate result is a vampire. And so it's actually believed that, in the Eastern European history, that some of these people actually suffered from uroporphyrinogen 1 defect. And so that type of porphyria does cause their teeth to glow. It does cause them to be very, very white. I don't know how they look at their urine or whatever, but yeah, so their bodily fluids would be red. And so it's believed that could possibly help lend credence to the whole idea of vampirism and the mythology behind that.
What about garlic?
I can't help you there, sorry. Or the bat.
For the bad.
So does this have to do also, like we were talking about, the blue people though?
Methemoglobinemia?
Yeah.
Right, that's something that's a little different where it stabilizes a different oxidation state of iron. And so it forms a type of, it's a porphyrin disorder where it forms, destabilizes the oxygen. And so therefore they do turn like a blue or silver kind of color.
Well, you explained vampires and Smurfs.
I know, this class rocks. That's one of the reasons why I love biochemistry. There is such a thing as vampire, and Smurfs are real. At least in Kentucky and West Virginia, the Smurfs are. No, I didn't say it.
In Arkansas.
Well, there's just a higher. The Fugate family is the one that is studied for hemoglobinemia, and they are from Kentucky and West Virginia area. So a lot of their relatives are blue people. And so they're the original Blue Man Group.
Wow.
Yes. I'll be here all week. All right. So now we're going to move on to creatine and phosphocreatine, because that's just a phosphorylated version. It's an important energy buffer used in your skeletal muscles, and so sometimes you hear people taking like creatine supplements, which, by the way, you need to be careful. You don't want to do it for too much. That's the kind of thing that you want to cycle on and off. Okay, and phosphocreatine comes from creatine, and creatine is formed from glycine once again, good old glycine, and arginine. However, it does require SAM, so SAM stood for what? It's also called adomed, but S? Adenosylmethionine. Okay. Yes. And so that's why technically it comes from glycine, arginine, and met, but met is just for the cofactor there. Methionine is for the cofactor. I would expect you to be able to rationalize and come up with the names for these enzymes. So if we just look at the first one, we have glycine and arginine coming together to make what's called guanidinoacetate, which is that's because if you look at from that nitrogen on, on up, which is frozen now. That's a guanidino group, which looks an awful lot, like the arginine. And then the acetate is just regular old acetate. So ornithine is what's given off, which we've seen ornithine. Ornithine can go on to do what in vivo? What cycle does it go to? It's the urea cycle. Okay, so what class or classes of enzymes could this be? There's two classes of enzymes I can think of, the broad overall classes. Is this like a phosphorylase or a phosphatase or anything like that? No. Is it like an oxidoreductase? No. It's not an aminotransferase. Oh, but you're really close. It is a transferase. It's an amino transferase. So the amidino is that NC double bond to the N. That kind of has like a hip hoppy feel to it.
So what's the difference between that and urea?
Urea has another atom. Right. So it's got another group on it. Now, methionine is used to make SAM, which is adomet. Okay, and it's given off as adohomocysteine. Remember, HCY is a 3-letter code for homocysteine. It's not one that I had you memorize last semester, but you should recognize it now, since we've seen homocysteine multiple times. It's just not an amino acid that is used to make a protein. It's used for this kind of reaction. I don't want to give it away. What's the big difference between guanidino acetate and creatine? It's a methyl group. So what class or classes of enzymes could this be? This is a methyltransferase. Whoops. Now I gave away the answer, and the final step, because I gave it away because I hit it too, so it's creatine kinase, which makes sense. You're only adding the phosphate at the expense of ATP, so it's creatine kinase.
So
is this part of the urea cycle since it uses like the arginine?
It is not, so let's see, how can I explain this? The cycles are not mutually exclusive. We've already seen that with the way that the citric acid cycle is directly linked to both the electron transport chain because of the fact that, complex 2 not fumarase, but the succinate dehydrogenases are the same. We've also seen how citric acid cycle is directly tied to gluconeogenesis since they have oxaloacetate as an intermediate. This is similar to that. So we have the urea cycle that's directly attached to the synthesis of phosphocreatine and creatine. And so it's not as cut and dried as where, you can only do glycolysis, or you can only do gluconeogenesis, or you can only do this. It's very complex.
So I thought creatine would be the final step. So why are you converting it to phosphate?
Phosphocreatine is the one that's used as a buffer system for the muscles.
Oh, it's not as
No. It's just that you can take the creatine supplement, and then your body's creatine kinase kicks in and converts it to phosphocreatine.
Does that help with what sore muscles? What does it help with?
Does anyone remember? We've actually seen what's some of the reasons. First of all, it's a buffer system, but secondly, what else does creatine and phosphocreatine do?
It was used to put water in your muscles or something.
Not water, but you're close. What was it like? It's transporting things from the cytosol to the holy of holies of the cell, the mitochondrial matrix. Remember, like the acetyl groups and acyl groups and fatty acids that you're going to use for making your muscles and for energy. And so you have to increase your creatine content. Or I should say don't have to, but if you increase the creatine content, remember it's the go-between in the intermembrane space.
So it helps to shuttle.
It helps with that shuttling. That we've seen back in chapter, whatever the mitochondrial chapter was. Okay. let's see, let me just double check, see how much is glutathione. So, next up is glutathione. And the 4 questions I want you to know, first of all, what is glutathione? We talked about it briefly, not so much, I don't think, this semester, but we talked about it really early on last semester. And what roles does it play, from where does it come, and how is it synthesized? Does anyone remember glutathione? It's a tripeptide. It's just 3 amino acids that are put together. But what is it important for?
Reducing the hydrogen peroxide, or making hydrogen peroxide from our own.
That's one possibility. I mean, it's one thing that it does, but what's its let's see. You use the keyword reduction because it's a redox reagent, but it's not just for hydrogen peroxide. What else does it keep reduced or oxidized as needed? Cysteines and disulfide bonds. It helps with all this, anything with the sulfurs, because this has a cysteine in it. Okay, and that's why glutathione can be a disulfide tripeptide, and so technically there would be 6 amino acids. Or we have ways to reduce it and oxidize it as need be, so that way it can form disulfides when you want to keep them protected and you don't want the protein to form an unwanted disulfide bond. It's utilized also with hydrogen peroxide and some of the other reactive redox components as well, since it can donate hydrogens or accept hydrogens.
So it forms and breaks down, or just forms disulfide?
No, it forms and breaks down.
Forms down.
Because there are times that whenever you're trying to fold the protein just right, that protein may have multiple cysteines, and you have to be careful because otherwise you could have 2 cysteines inside the protein form a disulfide bond that aren't supposed to. Or you can imagine having, let's say we've got a round globular protein. Let me see it. I could probably draw it better than do charades with it. But let's say we have this protein and it's got cysteines on the outside. The problem with that, if it's got exposed cysteines, is you can imagine 2 of them coming together in close contact, and now you've got 2 proteins that are stuck to each other that shouldn't be. And so glutathione can actually come in and I just do it as a G. That's not glycine, that's glutathione. But it can form, since it's tiny, it can help form protective disulfide bonds until you need to use that sulfur for something else or what have you. That's one way. And a lot of times they abbreviate the oxidized glutathione like this. We've seen it that way before, GSSG versus GSH. GSH is when it's fully reduced. When it's got a disulfide bond, it's oxidized. Well, it's gone to sleep. So, there we go. So, it's a modified tripeptide. It can form a disulfide bond with itself. It can also form disulfides with other things. It's present in animals and plants and even in some bacteria. I mean, it's just a really small tripeptide. And as I mentioned, the roles of these disulfides and properties of sulfur. Remember, sulfur is a great nucleophile and leaving group, but we have to be careful because of the fact that it can be so reactive in vivo. We've already seen how coenzyme A uses sulfur, glutathiones use sulfur, cysteines and cysteines use sulfur, homocysteine, methionine. So sulfur plays a very important role in vivo. Okay. So glutathione is a redox buffer. So for example, with hydrogen peroxides and some of these other things that you have to worry about. It's involved in maintenance of proper thiols and disulfide bonds for protein synthesis, and also to keep them from being aberrant protein, I'm trying to think of a good word for it. Protein plaque formation, like amyloids and things like that. And then it's also important for iron oxidation states for the hemes, because it can donate and accept electrons. So it helps to keep your hemes going well. This is what it looks like. And so it's the glutamate and cysteine. This is whenever it's oxidized. Glutamate, cysteine, and glycine. Okay, and it's the cysteine that has the reactive thiol. Now, whenever it's reduced, that cysteine would have an SH, and then you'd have two separate glutathiones. But notice it says gamma glutamate because it's not a normal peptide bond between the glutamate and the cysteine. Okay, so that's one way that your body can distinguish it from just, say, a regular protein. It's going to be oriented differently. And I can show you what it really looks like. Here, let me just go down. That's what it looks like overall. Okay, and we're not going to go into it today. No, we still have 5 minutes. But I just wanted to point out the whole idea of this gamma. The reason why it says gamma glutamate. If you notice, the gamma position is the terminal position, but glutamate has a carboxylic acid. And so if you think back to organic chem 2, you can attack two different carbonyl groups are possible to attack. You can attack the normal one to form the regular peptide, which would be on the alpha carbon, or you can attack the gamma one, which is the one that's on the end of the R group. And so this is the end of the R group that gets attacked. That's why it's called gamma glutamate, or technically gamma glutamyl, since it's the functional group that's added on. But the cysteine and the glycine are just normal. It's just the fact that this is a amide peptide bond. It's just not your normal alpha amide peptide bond. And so the next class will actually go through the steps, even though you probably figure it out since it's not particularly complicated. But are there any questions?